All right, let's get right into it. Welcome to this explainer on the wildly complex and honestly fascinating physiological mechanisms of urine formation and excretion. If you've ever wondered how the human body manages to relentlessly filter out the bad stuff while meticulously holding on to the good stuff, you are absolutely in the right place. We've got a really high-yield study guide lined up for you today. We'll be moving through 6 quick sections: evolution of waste removal, anatomy and the filtrate pathway, the countercurrent mechanism, hormonal regulation, clinical disorders, and then we'll wrap the whole thing up with a rapid-fire renal function pop quiz. So, Section 1, evolution of waste removal. Let's start by looking at the fundamental biological crisis that makes kidneys so absolutely essential in the first place: the accumulation of toxic metabolic waste. What's super interesting here is how the environment literally shapes biology. Aquatic animals, well, they have it incredibly easy. They just excrete highly toxic ammonia directly into the water around them because they have abundant water to flush it out. It's a process called ammonotelism. But as species evolved and moved onto land, water suddenly became this precious, scarce commodity. Terrestrial animals literally had to evolve to produce less toxic waste just to conserve water. So mammals, like us, excrete urea. Birds, reptiles, and insects take it even further, excreting uric acid as a paste with almost zero water loss. When you think about it, our entire renal system is basically just an evolutionary masterclass in water conservation. Moving on to Section 2, anatomy and filtrate pathway. To truly grasp how humans pull off this incredible feat of saving water while clearing out urea, we need to map out the structural hierarchy. Zooming in on the gross anatomy, the kidney is beautifully organized into distinct zones. You have the tough outer capsule, the outer cortex, and that deep inner medulla. And notice how the medulla is divided into these conical structures, the medullary pyramids. These pyramids act as biological funnels, directing the newly formed urine down into the calyces, which eventually pool together in the broad renal pelvis before making their exit out through the ureter. Now let's take a journey on a microscopic level. Inside those kidney zones are over 1 million individual nephrons. Track a single drop of fluid, which we now call filtrate, with me. It starts at step 1, where blood is filtered under really high pressure in the glomerulus and caught right in Bowman's capsule. From there, this protein-free plasma twists its way through the proximal convoluted tubule, takes a massive dive deep down into the loop of Henle, comes all the way back up to the distal convoluted tubule, and finally drains right into the collecting duct. And that massive dive brings us to Section 3, the countercurrent mechanism. As the filtrate travels that path, it hits a famously intricate physiological structure deep in the medullary tissue. This is the countercurrent mechanism. Honestly, it is the absolute star of mammalian renal physiology. It is defined by the parallel arrangement of the loop of Henle and its surrounding capillary network known as the vasa recta. The filtrate flowing in the loop and the blood flowing in the vasa recta literally move in opposite directions, a true countercurrent. This ultra-precise setup creates an intense concentration gradient in the medullary tissue, which is exactly what allows human kidneys to produce urine that is nearly 4 times more concentrated than the initial filtrate. The absolute magic of this mechanism lies in opposing permeabilities. As the filtrate travels down the descending limb, water is aggressively drawn out into that highly concentrated medullary tissue. But the electrolytes, they're trapped inside. So the filtrate gets super concentrated. But then it turns the hairpin corner at the bottom and heads up the ascending limb. And the rules completely flip. Suddenly, the ascending limb is totally impermeable to water, but it aggressively pumps those electrolytes back out into the tissue to maintain that intense gradient. It's just a brilliant, continuous, self-sustaining loop of concentration and dilution. Which brings us to Section 4, hormonal regulation pathways. Because of course, this system isn't just static. Your body's hydration levels and blood pressure are fluctuating constantly, meaning this whole process requires highly dynamic regulation. A perfect example of this dynamic regulation is the renin-angiotensin mechanism, a vital cascade that kicks in completely automatically. Imagine your glomerular filtration rate, or GFR, drops. Instantly, special cells in the juxtaglomerular apparatus, the JGA, detect that drop and release an enzyme called renin. Renin is the first domino. It eventually leads to the creation of angiotensin II, which is a very powerful vasoconstrictor. Angiotensin II then signals the adrenal cortex to release aldosterone, which practically forces the kidney tubules to reabsorb sodium and water. The end result? Your blood volume and blood pressure are pushed right back up to normal levels.
At the exact same time, you've got this fascinating hormonal tug-of-war happening. If you're dehydrated, the hypothalamus releases ADH, antidiuretic hormone, which opens up water channels in the late tubules to save every drop of fluid it can. But what happens if your blood pressure gets dangerously high from all that water and sodium retention? Well, your body has a built-in brake pedal called ANF, released straight from the atria of your heart. Atrial natriuretic factor causes vasodilation, keeping that whole renin-angiotensin cascade safely in check.
Section 5. Clinical Disorders and Organs. Let's take all of this amazing physiology we just covered and bridge it to the clinic, looking at systemic failures and the other auxiliary organs that pitch in. So the kidneys usually get all the glory, but they definitely don't work alone. Your lungs are actually removing a massive 200 milliliters of CO2 every single minute. The liver is busy secreting degraded hormones and cholesterol into your digestive waste. And you can't underestimate the skin. While sweat is primarily there to cool you down, your sweat glands are crucial secondary systems for eliminating water, sodium chloride, and even traces of urea and lactic acid. Even your sebaceous glands are pitching in by eliminating sterols and waxes via sebum. The skin is incredibly dynamic. But clinically, when the primary system, the kidneys, actually fails to clear out that urea, patients develop a really dangerous condition called uremia. This is a highly toxic state of accumulated nitrogenous waste, and at this point, survival relies on hemodialysis. This is where an artificial kidney literally steps in. It uses a concentration gradient across the cellophane membrane to wash the toxic urea right out of the patient's drained blood before safely pumping it back into their body. You can also spot systemic issues just by looking at what's spilling into the urine. Because the proximal tubule is supposed to reabsorb 100% of essential nutrients, seeing glucose in the urine, known as glycosuria, especially alongside ketone bodies, is a massive hallmark diagnostic indicator of diabetes mellitus. The kidneys are literally showing you what's wrong systemically. All right, Section 6, Renal Function Pop Quiz. To really lock in this high-yield review, let's switch gears from passive listening to active recall with a quick rapid-fire quiz based on the core textbook exercises. Think fast. Question number 1, true or false? Micturition, which is the clinical term for the release of urine, is carried out by reflex. That is true. As the bladder gradually fills up, stretch receptors on its walls fire signals to the central nervous system. The CNS then sends motor messages right back down to trigger the smooth muscle contractions that actually release the urine. Okay, next question. Think back to the very start of the nephron. True or false, a completely protein-free fluid is filtered from your blood plasma directly into the Bowman's capsule. True again. The high pressure forces your blood through these intricate slit pores created by cells called podocytes. They act like a microscopic sieve, ensuring massive protein molecules stay safely in your bloodstream, while the watery, nutrient-rich plasma enters the tubule. Last question, and it's testing that famous countercurrent mechanism we talked about. Fill in the blanks. The ascending limb of Henle's loop is blank to water, whereas descending limb is blank to it. Did you remember the flip? The ascending limb is completely impermeable to water. It's just pumping out salts. Meanwhile, the descending limb is highly permeable to water. That specific opposing dynamic is quite literally the foundation of how mammals survive on dry land. Let's leave on this wild thought from the source text. Your kidneys produce an astonishing 180 liters of glomerular filtrate every single day. 180 liters. Yet you only excrete about 1.5 liters of urine. That means a staggering 99% of the fluid passing through your microscopic nephrons has to be actively reabsorbed back into your body just to keep you alive. When you sit back and consider this relentless, silent, microscopic balancing act happening inside you right at this very second, how does understanding your renal physiology change the way you view the true value of a simple glass of water? Keep learning, stay curious, and I'll catch you on the next explainer.